everyone, and welcome to our interview series marking the 200th birth anniversary of the father of modern-day genetics, Gregor Mendel. I'm Astha Vatsai, a PhD student in Dr. Vinod Skaria's lab, and today we have with us Professor G. Kumaresan from Madurai University. Sir is the head of the Department of Genetics at the Unit of Excellence in Cancer Genetics there. Sir's areas of interest include cancer genomics and targeted therapeutics, with special focus on breast, gastric, and hepatocellular cancers. Welcome, sir. It's a pleasure to have you here with us today.
Yeah, thank you. Thank you very much for this opportunity, actually.
Sir, you made important contributions to the field of genomics and cancer biology. Could you briefly discuss with us what your current areas of interest with respect to your work are?
Yeah, currently we are working on three different aspects. One is cancer genomics. So mainly we are working on breast, gastric, and liver cancer. So what we are trying to understand is the biology of cancers by using genomics approach. So we are using various genomic platforms and trying to understand the biology of gastric cancer, breast cancer, and liver cancer. So by this way, we are trying to understand the molecular dysregulations because basically cancer is a kind of basic alteration in the biological pathways, signaling pathways, and molecular dysregulations. And we are trying to delineate those dysregulations by using genomics approaches. And ultimately, we are understanding various pathways, dysregulations involved in cancer. That is one part. And second thing is we are trying to, because the major problem in cancer today is, we know the biology to some extent, but the problem is heterogeneity because the one drug may not work for all the patients. So the patients needs to be stratified, which drug would be suitable for one individual. So the major problem is heterogeneity. So we are trying to delineate the heterogeneity. For example, we are working on the concept of like gastric cancer can be subdivided into multiple subtypes because as of now in clinically maybe 2 or 3 subtypes are known, but we are working on the possibility of categorizing beyond that. So for example, a cancer could be divided into maybe 10 subtypes. So now in 10 subtypes, we know the molecular biology, we know the dysregulation, we know the signaling pathways dysregulated individually in 10 subtypes of a particular cancer. Now, this is a step towards precision medicine or targeted therapy or personalized medicine, whatever you call. So that is the ultimate goal with which we are working on. So that's a major area in which we are working on. The second area is we are trying to understand the real molecular targets of drugs because we are using a lot of drugs. I don't think humans can live today without any chemical modifiers or like drugs. But for many drugs, we know the mechanisms to some extent, but still we do not know the 360-degree molecular targets of all these drugs to the fullest possible potential. So we are trying to understand various molecular targets of the drugs which are already in clinical use, and also some drugs potential candidates. And so the drugs can be used in a very safe way, at least in the future. So we are trying to delineate the molecular targets of the drugs. So that is the second area in which we are working. And recently, we are also into the ancient genomics of Indian archaeological skeletal remains. So in Tamil Nadu, there are some 7 excavation sites, and from those sites we have access to a lot of skeletal materials of 2000, 3000 years old, and we are trying to do some genomics from those samples and then trying to understand their population genetics and so on. These are the major areas in which we are working on.
So that's fascinating. That's I think taking population genetics to the next level, going back in the past. Sir, how do you think Mendel's discoveries have impacted your work so far?
Yeah, basically, Mendel started with basic inheritance of the factors at that time, and then so that is a base. We started that is basically ABC in genomics actually. Without that, we could not have any idea what we have today because we are talking about genomics. But the only difference is at that time it was talking about the inheritance of the factors, but now we are talking about the the whole genome of 3 billion bases and so on. So it really basically it is from Mendel's discovery, and then subsequently now we are into all possible because now it's entirely different actually. In population genetics, we talk basic thing is inheritance, the basic inheritance of the factors. But without that, I don't think we can think of or even even imagine of whatever the understanding what we have today in genomics. So in that context, it's really it's a base actually. Without that, nothing is possible.
Absolutely, sir. Sir, definitely for researchers, but do you think studying genetics is also important or beneficial for the common man?
Yeah, of course, because now we are talking about so now for many diseases we have drugs. That is a development in the last 3 4 decades. But the next stage is like we need to have precision medicine. So that is basically based on the genetics of the individual, because some drugs may not work for one. Sometime back I was talking about the heterogeneity in cancers. So
Yes.
basically the heterogeneity is because of the genetic changes. Now it may not be like exactly genetic, but sometimes it's somatic changes also. But basically those are the changes and inheritance from one cell to the another cell. So that is a base. Without genetics, like we talk about like modern genetics, genomics, like personalized medicine, targeted therapy, everything, the basically it is everything is an advancement in the basic classical genetics. So that is really going to be helpful for the modern life, because the drugs we are now, what people are trying to do is, yeah, drugs are already there in plenty, but we need to link them with individuals. So in that context, like we have to link a drug with a particular genotype. So basically it's a genotype to phenotype correlation, but in the context of drugs. For that, the basic knowledge is from genetics. And so that is definitely is being, already in many diseases people started using that. But I'm sure that in the next decade, for the coming years, definitely like personalized medicine is going to be play a big role. And in that basic is genetics actually.
Right, absolutely. Sir, your work is very translational in nature. It directly impacts people, patients. Do you think the knowledge of Mendelian genetics would actually help shape public policy, healthcare policy in some ways or the other?
Yeah, definitely, because already people are doing that actually. And in India also we have the like policy and everything is like maybe it's in the early stage, but things are evolving. And because now ultimately people are thinking about the sequencing the genome of every individual. Okay, that is the definite like the others, the ultimate development of the genetics. And for that obviously there are a lot of but most often the policies are related to the ethical aspects, not because we don't have any other issues actually. So definitely the policies would be evolved, but I think that is more of related to the ethics in my view. Because otherwise genetics is always being useful, and I don't think there is any restriction in because there are some social issues might arise because of that. So ethical considerations are more important. So in those contexts policies are required, otherwise it's fine.
Right, right. So you mentioned that you were studying DNA from 2000, 3000 years ago. Now, population genetics definitely relies, population genetics and genomics definitely rely on genetics a lot. So is this project of yours also linked with genetic studies? Are you going to be
Yeah,
performing this?
obviously, this project itself is ancient genomics project actually. So genomics is nothing but like is a technology what we are using
Right.
to like uncover genetics. So genomics is nothing but is a kind of technological advancement in genetics. So obviously, it's a genetics project. So we are trying to understand the inheritance, like how it happened in the past 2000 or 3000 years old, and then how it is linked to the present population. And this is just to get some understand the history by using genetic methods or genomics methods. So obviously, like if you probably we are forgetting about genetics actually, and then we always talk about genomics and all those things. So basically everything is from genetics. And thanks for this opportunity. At least you got given opportunity to think about everything, because in day-to-day life probably we are not talking about, but I think, as I said, ABC is genetics. And then we are building up. I think the science has grown a lot by using those basic knowledge, and I'm sure that is going to be beneficial to the, I think already it is being beneficial to the society.
Right.
And it will reach different dimensions, and it will be extremely useful in different aspects
So
in all
what
the.
Absolutely. So that brings me to the final question. What do you think is the way forward? How do you see things progressing from now on with respect to genetics, with respect to genetics in your work specifically? How do you think things are going to progress now?
Yeah, actually, as I said, personal genomics is a kind of way forward, and then many countries already they have started. And now I think sequencing the whole genome of an individual will not going to cost more than 50,000 rupees or so. And so personal genomics is realistic. It's not, it is a realistic possibility. So eventually, maybe like 10 years from now or so, I'm sure that many of us would be having our own genome sequence in ready. So the thing, what is really necessary is we need to have developed parallel computational methods and then understanding methods, and that is one side we need to really concentrate. And second thing is, and many countries are working on those to be used by the individuals, because for biologists it may be easy to understand, but even common man also should be able to understand this, because once you have your own genome sequence, and you should be able to understand that, and there should be some, like, methodologies evolved for that, because ultimately everybody needs to understand, clinicians needs to understand, and, like, some other policymakers needs to understand. So that is one thing. And the another one, you also highlighted the kind of some policies, because once we have all those things, we may have, like, unexpected issues possible, which may stem from different sectors, and then which we may not probably even thought about.
Yeah.
So we also need to have stringent policies, and then insurance policies. All those things are the problems in the other countries, because once you have the personalized medicine, and then those kind of things needs to be evolved parallel, and we need to have the capacity building, and then we need to have a very kind of brainstorming, and then we need to have a legal protection for the scientists in that context, and all those things for the individuals, clinicians, and everything. Those
Yeah.
are the areas which we need to concentrate. And while coming to our work, we are working towards, so once a patient comes to a clinic, and we need to, like, do the transcriptome analysis or genome analysis, and then try to say that, okay, this individual belongs to this subtype of maybe a gastric cancer, and these all are the dysregulations in these individuals, and then directly connect that to a particular drug, or maybe a set of drugs. And so it's a kind of personalized medicine, and I believe it is possible in, like, at least in next 5 years or so. So we are working with that technology, with that methodologies. And then, but obviously the challenge is, like, I am talking this from the lab, and this has to be in translational context. I think probably there are, like, several hundred yards are there between what I think and then when it comes to practical usage. But I strongly believe that it is possible. So we have the knowledge, and then obviously we can translate. So again, as I said, like, once we have the mechanisms of the drugs are well known, then we can directly connect them with a subset of patients or with the individuals. And so it is towards personalized therapy and for cancers. And I think same kind of things are possible for other diseases also to some extent. So maybe for diabetes right now, like, maybe 2-3 classes of drugs, but why don't we, like, stratify the patients into multiple subtypes, and then treat individuals with the different categories of drugs. So ultimately, mainly in cancers and also possibly in other diseases, we are trying to develop personalized, stratified, and targeted therapies. And so I believe it is possible. Yeah.
Absolutely. So, someone who's studying familial cancers herself and who's from a computational biology lab, I completely agree with all the points you just made. Thank you so much for sparing us the time and talking to us, sir. It was a very, very interesting conversation. Thanks for being
Yeah,
here with us.
thank you very much. Thank you very much for this opportunity. I'm sure that it will be useful to the students and public.
I'm
Thank
sure,
you
sir.
very much.
Thank you, sir.